Hello, everyone. Thank you for coming to my session today. Today, I'm going to be talking about maintaining academic rigor in online labs. And so how to optimize this learning environment for labs, which I want to start off by just reviewing what the typical lab environment looks like. And so as you guys are aware, when students are in a lab session, they're working in groups, completing experiments and analyzing data. And then they're also navigating their way through the scientific method as they're completing these experiments, formulating hypotheses, questions, and so on and so forth. And in addition to that, they're practicing and mastering hands-on laboratory techniques. And I think we can all agree that when we move into an online setting, a lot of these qualities of a typical lab environment are severely compromised. So it's challenging to get students to work in groups. They're not able to, complete experiments necessarily or analyze data as well, and work their way through that scientific method. And then last, and I think what's most important, is that students start to lose this ability to practice and master laboratory techniques. And this can also be, detrimental for when they move up to higher level labs, or if they want to pursue a career working in research or something like that. These skills that they learn in a lab are really, really important. But this doesn't have to be the case. Over the last year and a half with COVID, I've been really fortunate to have some time and devote to figuring this out and how to make the typical lab environment present in an online setting. And so I have listed here a couple of strategies that I have, developed and used, and I'm really excited to share them with you. And hopefully, you can maybe walk away from this session with having a couple ideas of how you want to make your online lab environment more of a typical lab environment for your students. So the first one I want to talk about is generated data with supplemental videos. And so in this experiment, we're demonstrating the scientific method / experimentation. And the two examples that we use this one for are the corn genetics lab that we use and the Drosophila lab. So both of them are looking at Mendelian genetics. The second strategy that I've developed is lab kits. So right now we use 2 of these 4 lab kits in the Biology 101 labs at the University of South Carolina. The pH and buffers and the photosynthesis have been highly successful, which is really, and we're piloting the diffusion osmosis lab this semester, and then hopefully the enzyme lab kit in the spring 2022. These take a lot of time to develop and, practice and also, distribute to all the students. So these, I usually am trying to roll them out slowly. So we did pH and buffers and photosynthesis in the spring 21, and now we're starting to add more and more. The last 2, which I'm going to spend not as much time on today, but I'm happy to address in the question session, or if you want to send me an email about these, I'd be happy to help or provide some of my resources. This would be the guided assignments, which is, we have 2 examples for these, which are the lab summary quizzes. And in these, we have an abstract style of writing, and it helps the students to learn how to utilize the scientific method and encourages this, lab report writing style, and gives them a this. The second one is our interactives. So we have 3 interactives that we use throughout the semester for Biology 101 labs here, one being on macromolecules, one looking at mitosis and meiosis. And then the third one is more of a tutorial style where we walk through with the students how to use Excel. So these actually were developed with Hayden McNeil, and they're really, really fantastic. So I would be happy to, share any of those or what we do in these interactives, but these are basically assignments in labs that they can do on their own, and it helps them master this content through walking through this interactive PowerPoint where they're clicking on things. It's really nice. And then the last thing that we do are case studies and these group projects. So, I want to have a little bit of a disclaimer here. Not every single one of these strategies is going to hit every quality of a typical lab environment. But what I think is really cool about these is that we're able to hit usually at least 1 or 2 of these typical lab environment qualities in these, new online teaching lab styles. So I want to start off with going over the Drosophila one. And so in this one, we have the students generate data, and we are using instruction through some of these supplemental videos that we've created. So I'm going to share a few of the videos today, not all of them for the sake of time, but just kind of to show you what we're doing and how we're trying to make the lab more realistic to them. So they're understanding, how Drosophila vials are set up, or how we're setting up these culture tubes so they can look at the F2 generation. And so throughout this exercise, what the students are going to be doing is reviewing Punnett squares. They're going to theoretically set up the cross of the F1 generation, and then in 2 weeks' time, they would observe the F2 generation. And then in this experiment as well, we're having them, based on the F1 generation, predict what the parental generation was, and then also learning how to do chi-square analysis. And so in this lab exercise, I'm sure you guys are fairly familiar with this kind of setup when you're looking at Mendelian genetics crosses with Biology 101 labs. But we, first review how to set up Punnett squares and how to complete this and determine what their expected F2 generation is. So we have the gametes of the F1 going across the top and bottom, or top and side. And then, we walk through with them how to fill in this Punnett square to get their predicted 9:3 to 3:1 ratio. And so in this experiment, we're looking at 2 mutations that are recessive. So, one would be the vestigial wings, or the, kind of shriveled wings, and then the ebony body color. And so what we do is we walk through with the students what would happen in a normal lab environment. So we have this cross of flies, which is the F1 generation here. And then, we're showing them at this point in this video how to anesthetize the flies. So I'm going to play this video. It's pretty short, just showing, how to do this procedure.
Yeah.
Okay, so what you can see from it, it's very short, very simple, but it's allowing them to see a couple things. First off, they're able to see, okay, what do these fly tubes look like when they arrive, from the company we order? How are we able to, get these flies to go to sleep so that we can, set up breeding crosses, and then also examine our flies, once the 2-week time period has ended. This also, I think, in an online setting allows us to kind of speed up the process because we don't have to wait that 2 to 3 week period. So, we'll walk through, okay, this is how we're going to anesthetize them. We're going to, set up our crosses, and then 2 to 3 weeks later, we're going to observe those flies. And so we're able to just kind of fast forward 2 to 3 weeks and observe the F2 generation.
Yes.
Okay, so usually it takes about 5 minutes for the flies to fall asleep using fly nap. And during that time, we'll explain to the students how to determine the gender of these flies. So, the males usually have a black abdomen and these sex combs on their arms, whereas the females just have a striped abdomen. We have a couple more examples of how to do this. It's usually easiest if the fly is on their back, and you can easily see that this is the male and then this is the female. So this is usually a very challenging skill for students to learn. So going over this, I think, is really beneficial to the students just to kind of get used to this and looking at flies through their experimentation. And in addition, in our lab, we typically do a sex-linked cross as well. So reviewing how to determine the gender of the flies is really, really important for them to be able to count their flies and analyze their data. Okay. And then typically in a lab, while those flies are falling asleep, we would be setting up their culture tubes. And so in this video, we're showing how to do that and what the steps are for that. So I'm going to play this video as well. It's very short, but it's nice because it shows them, what we would be doing in the lab. Awesome. So this is a very simple procedure for how we set up our fly crosses. And then at this point, this is when we would, open up the flies and, distribute to the students, allow them to observe them under the microscope. Okay, and so because we're not able to look at these actual flies, I've had some grad students over the last couple months working on these files. And so what they were generating is several slides of PowerPoints looking at different flies. And so they were did some editing on these to make them have vestigial wings over here, for example, or ebony body color and that double mutant over here. And so what we do is we'll assign students to work in groups of 2 to 3, and we'll move them into breakout rooms, and they're going to count the number of flies in each of these categories. So wild type body and wings, ebony body and wild type wings. Let's see, where's the vestigial? Wild type body, vestigial wings, and then this double mutant here. And what we end up doing is we count all these numbers and we'll put them into a Google sheet where they can enter in all of their data and then proceed through doing a chi-square analysis, seeing if their observed values or counts of these flies match what you would expect, which would be that for this cross, the 9 to 3 to 3 to 1 ratio. So I know it was kind of a quick snippet of what we do for this lab. We have a couple other videos for the Drosophila lab that I'm happy to share if anyone is interested. But the goal of the supplemental videos with the generated data is to give them as realistic of a view of some of these labs as possible. And more importantly, getting them to, count flies and observe the data. So I think this kind of provides a wholesome view of doing the experiment, what goes into that, and then also counting the flies and determining these different mutations. Okay, the second one I want to move into is looking at our custom lab kits. And so these are actually made by myself, as well as a couple of work study students that I have that helped me put these together. So I want to shout out a thank you to them because it would really not be possible without all of their assistance on these. So I've decided to share our pH and buffers one because it is the most simple. So it'll be, relatively easy for us to walk through. The photosynthesis one is also pretty simple and really fun. So the students can do this outside or in their house or wherever. But they're both really, really nice experiments that are easy to recapitulate at home. Okay, so for this study, just to give you guys a little bit of background, we're studying the pH scale. And so what we're going to be using in this study is lemon juice as our acidic solution. And this is what we're going to be using to modify our solutions. In addition, we're looking at the impact of lemon juice on solutions. So one of them being a buffer, which is a substance that minimizes change in the concentration of hydrogen or hydroxide ions. And so what this means is that by adding an acidic or basic solution to a buffer, the pH will remain fairly consistent. And what this means is that buffers are able to accept protons right here where the star is, and they're able to donate protons here. And so this allows the pH to remain relatively stable when you're adding something acidic, such as lemon juice, which has a high hydrogen ion concentration. Okay, and so the experimental design for this one is very, very simple. What we have is our bicarbonate solution, and that when we're adding lemon juice to the solution, we're going to be studying how the pH is changing. So it's going to accept hydrogen ions and prevent the pH from decreasing or becoming more acidic. Okay, and so in this lab, we have two objectives. The first being to determine the pH change of water and a bicarbonate solution through the addition of drops of lemon juice. So they're formulating their hypothesis based on how they think lemon juice is going to impact the pH of water and bicarbonate. And then the second one, which is a little bit of more of a fun experiment, is identifying an unknown solution. So this one, we either can put, water, a really strong buffer, a weak buffer. So this kind of leads to, a little bit of open-endedness on, the instructor's end on how they want to navigate this. I usually just make either a really weak buffer or a really strong buffer, but the weak buffer is interesting because it'll kind of start ending up in the middle because it'll reach its buffering capacity earlier than our typical buffer. Okay, and so this is all of the equipment that is involved in each lab kit. So we have 3 glass vials, 4 plastic droppers, 4 plastic tubes. And so these are just 15 mil tubes that will have water, bicarbonate solution, lemon juice, and an unknown solution. So we provide them with 3 times the amount of reagents they would need. And this allows them to repeat the experiment if they need to, or if some spills, they'll have plenty. And then a long piece of pH paper that they'll then be able to break into several pieces. And so what you can see from this experiment is that the supplies that are required are really, it's a really short list and very simple. And so these are things that are easy to put together and easily distributed to your students. In addition to that, they're, very inexpensive and safe for the students. So there's nothing in here that's, biohazardous or dangerous for them. Okay, and so what we do for this lab is we'll have, somewhere for, our lab kit, somewhere between 8 to 10 steps, walking them through how to do this experiment. And what my TAs will do is go step by step with their students with their camera on, walking through, okay, make a table like this, for example, and tear 18 strips of pH paper and put them into these white squares. And so what's really nice about this is that even though we're online and it's harder to receive help from a TA, the way we have this set up is that there's step-by-step process where the TA is not going to move on until everyone has accomplished this first step. So it allows the students to receive the help that they would normally get in a lab. And students can turn on their cameras, turn on their mics, ask questions, get some assistance when needed. So the first step of this one is to create a table, put the 18 strips of pH paper onto the chart. Then we'll take out our 3 glass tubes and label them with either a Sharpie or some tape or a marker, water, bicarbonate, and their unknown solution. And then they're going to add 5 mils using their plastic pipette droppers into the water, 5 mils into bicarbonate, and then 5 mils into the unknown. At this point, they're going to measure the initial pH of their solutions by placing a few drops, usually 1 to 2 is sufficient, onto their pH paper. From there, they're going to move on to step 4, adding 2 drops of lemon juice to each vial, and they're going to mix the solution by pipetting up and down a few times. And so what's really cool, I think, about these experiments is although they're very simple, we're teaching them some really important skills. So for example, their water pipette needs to only be used for their water solution. So they're going to, add 2 drops of lemon juice, put that pipette dropper down, pick up their water pipette, mix it up and down to get that solution homogenized. In addition, we're also showing them what 1 mil looks like, where it ends on the plastic pipette dropper. So a lot of kind of smaller skills that, researchers would take for granted that really need to be learned in the Biology 101 labs. And then what they're going to do is measure the pH again by placing a few drops on the pH paper under column 2 here, which is 2 drops of lemon juice. And then what they're going to do is continue this process until they reach a total of 20 drops of lemon juice. So now total number of drops of lemon juice being 5. So they're going to add 3 additional drops to all 3 of their solutions, pipette these up and down really well, and mix up their solutions. So at the end of this, they should have all 18 strips of paper covered with a couple drops of lemon juice of their solution with lemon juice drops accommodating all of these columns. At this point, they'll then refer to the pH strip legend, which we have here, and they will record the pH at each of these squares. So let's say this one's 7, and then recording the pH changing as we go through this process. This data will then be collected into a table. And so we'll instruct them to open Microsoft Excel and work through creating a table similar to this and inputting the pH of their solutions going down as we increase the number of drops of lemon juice. Finally, we'll have the students create a graph. We'll do a scatter plot looking at how the lemon juice is affecting the pH of water and the bicarbonate. And then for this one, for example, you can see that the unknown solution was also a buffer. And so at this point, they can, compare their data to their hypothesis. So, for example, a lot of students will say the pH of water is going to drop significantly, whereas the pH of sodium bicarbonate, which is a buffer, is going to have a mild reduction in pH due to the buffering capabilities of sodium bicarbonate. So at this point, they can look at their data and see if their data supports their hypothesis, which is really, a powerful part of going through the scientific method. And so I just kind of want to highlight some of the really, important aspects of this lab that are used. So they are getting this hands-on experience, and they're learning to become comfortable with using pipette droppers, measuring accurate volumes, determining the pH of solutions, and comparing that to a legend, collecting data. And honestly, something that students struggle with the most are working in Microsoft Excel. So creating these tables and figures and then comparing their results to their original hypothesis. All right, so we do a couple extra lab kits as well. So I had mentioned the pH and buffers lab kit and photosynthesis are ones we currently are actively using, and we're test running the diffusion lab kit, and then hopefully in the spring we'll get the enzymes one down. And so what you can see from this lab kit is that it's a little bit more intricate in the materials that are provided. We make two 50 ml tubes of 2% NaHCO3 to be the carbon dioxide source for photosynthesis. We have an empty 50 ml tube that would be used for making their dilutions. So for this experiment, they're using 0.2 NaHCO3. So we walk through with them how to do these dilutions. We also have two Petri dishes to observe their two different conditions. So they'll cut up pieces of spinach, use the syringe, which is down here, to evacuate the oxygen from them so that they sink to the bottom of the Petri dishes once the solution is added. And through these two Petri dishes, we're able to create two different conditions. So the first one, for example, is looking at light versus dark conditions. So for our dark conditions, we'll cover this Petri dish with a piece of foil. And then there's a couple other, we have them do two experiments. So there's a couple options for what they can do for their second environmental factor. And we allow the students to select this one. So when they come to pick up their kit, they can either grab a piece of cotton gauze to look at light quantity. They have a color filter to look at light quality, and then additional tube of 2% NaHCO3. So looking at different carbon dioxide concentration. So comparing 2% to 0.2%. What I really like about this kit is that it allows them to learn a little bit about dilutions, which is a skill that a lot of students struggle with. And this is actually something that they don't do in the face-to-face lab. So we're trying to limit the amount of supplies that we're handing out to the students, just so we don't have to give them, 20 tubes of NaHCO3. We made it a more concentrated solution, and they're going to use this empty 50 ml tube to make their dilutions. So we first off walk through how to do this dilution. So we're going to be using 0.2% NaHCO3. You will therefore need to dilute your solution of your 2% into the 50 ml tube. And so at this point, this slide is usually a little more edited. So we allow things to pop up. So walking through with them, how much 2% NaHCO3 and how much water do you need to put into your 50 ml tube to create a 0.2% stock? And so the students will, we'll give them a few minutes to work on this and, provide help and feedback. But, hopefully by the end of this, we know that it's a 10 fold dilution. So we will have 5 mils of 2% NaHCO3 with 45 mils of water. And so whenever they're using their solution for this experiment, they're going to need to use this dilution. I'd quickly introduce these lab kits as well. The enzymes one has been a little bit challenging just to get practical reagents and supplies out to students. The one that we've had the most success with so far is either looking at catalase enzyme, looking at O2 production, or zymase, which is looking at CO2 production. So this lab is very, very nice in person. It's just a little bit difficult to nail in an online lab kit setting. And then the second one is diffusion. So the movement of glucose and starch across a semi-permeable membrane. So this one is pretty simple for us to distribute out to students. So I'm excited for this one to get going in a couple weeks. I would like to take this time if anyone has any questions. Thank you for being here, and I'm really honored to share my work with everyone.